Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine this incredible journey, right? A microscopic race against, well, unbelievable odds. Millions start, but only one can finish. For so many couples, this unseen struggle, male infertility, is a really tough reality. But what if we could actually peek inside that tiny cell that starts it all? Understand why that journey sometimes stalls before it even begins. Well, today, that's exactly what we're doing. Taking a deep dive into some fascinating research that's starting to, you could say, break the code of how sperm move. We're focusing on a new study out of the University of Osaka, published in Nature Communications. It's identified two really key proteins. Our mission: to pull out the crucial insights for you, understand what makes sperm swim, what goes wrong when they can't, and what this might mean for, future help with male infertility.
Yeah, it's such a critical area. Like you said, fertility challenges are immense for many people, and pinning down the exact cause, especially on the male side, has often been, well, really difficult. We know sperm motility is vital, obviously, but how the specific molecular mechanics has been a bit of a black box. This study, by naming these proteins and figuring out their jobs, it gives us a really important piece of that puzzle.
It's like getting the schematic for the engine.
Exactly. A clearer blueprint for male fertility. We're getting closer to understanding how it all actually works at that tiny level.
Okay, so let's unpack this then. Most people picture sperm, the tadpole shape, head and a tail. But that tail, the flagellum, it's not just some simple little whip, is it? It's more like a tiny submarine's propeller. Super powerful, incredibly complex.
That's a great analogy. And it really is complex. Inside that flagellum, there's this highly organized skeleton called the axonum.
The axonum.
That's the core engine, basically. It provides the structure, the machinery for movement. And within that, you have these really intricate T-shaped structures. They're called radial spokes.
Radial spokes, okay.
Right. And these are absolutely essential. They control how the flagellum actually bends and moves. Think of them like tiny gears or levers inside the propeller, regulating that precise wave-like motion.
So they allow it to swim purposefully, not just flail around.
Precisely. Without those radial spokes working correctly, the whole propulsion system just doesn't function properly.
And researchers have kind of suspected for a while that problems with these spokes are linked to male infertility, haven't they?
They have, yeah. It's been a known weak spot, you could say. But identifying the specific proteins doing the building, orchestrating the whole thing, that's been the tricky part. This study is exciting because it really zooms in on two specific proteins, gives us some concrete players.
Okay, so let's meet the first key player. It's called cilia and flagella associated protein 91. Let's maybe call it CFAP91 for short.
Yes, CFAP91. It's a known component of those radial spokes we just mentioned. And it had already been sort of implicated in human male infertility.
Implicated, but not fully understood.
Exactly. We knew it was involved, but how it worked, its precise function in making sperm work, that was still pretty unclear.
So how did the researchers figure out its specific job?
Well, they used a really standard but powerful technique. They engineered mice that couldn't produce this CFAP91 protein. These are called knockout mice.
Right. Remove the protein, see what breaks.
Precisely. See what happens when it's missing. They then looked very carefully at the sperm from these mice, their shape, their movement, and crucially, to really confirm it, they then put the protein back into some mice, re-expressed it to see if that fixed the problem.
That back and forth sounds pretty thorough.
It is. It really strengthens the conclusion that the protein itself is directly responsible for the effects you see.
Okay, so they took out CFAP91. What happened to the sperm? What did that tell them?
The results were stark, really clear-cut. First off, the mice were completely infertile.
Completely,
which right away tells you this protein is essential. Second, their sperm had major problems with how the flagellum, the tail, formed.
So not just problems swimming, but the tail itself didn't even build right?
Exactly. The tails were shorter than normal, and they were totally immotile, couldn't swim at all.
Like that submarine without a propeller.
Right. And when they looked closer, with microscopes, they saw widespread structural issues. The whole internal skeleton, the axonum, was disorganized. Other vital parts were messed up too. But the clincher was, when they put CFAP91 back in, fertility was restored.
Okay, so no doubt then, CFAP91 is indispensable.
Absolutely.
So it sounds less like just a part in the structure and more like the master builder, the one laying the foundation.
That's a perfect way to describe it. The study showed CFAP91 acts as an essential scaffold.
A scaffold?
Yeah, particularly for assembling one specific type of radial spoke called RS3. Without that initial scaffold, the rest of the structure, the whole flagellum, just can't be built correctly. And what's really interesting is that this protein, CFAP91, it's found across many species, including us humans. It's evolutionarily conserved.
Meaning it's fundamentally important, biologically speaking.
Exactly. It's been doing this critical job for a very, very long time.
Okay, but the story has another chapter, right? They didn't just stop at CFA91. How did they find the second protein?
Right, so they wanted to know who CFA91 hangs out with, essentially, in mature sperm. So they used this really neat technique called proximity labeling.
Proximity labeling. Sounds like mapping the neighborhood.
Kind of. Imagine tagging CFAP91 with a tiny chemical marker. This marker then sort of paints any other proteins that get physically very close to it within about 10 nanometers, which is incredibly close.
Wow.
It gives you a snapshot of its immediate molecular neighbors in a fully formed sperm cell.
Like finding out who lives right next door.
Exactly. And doing this, they consistently found another protein right next to CFAP91 in these mature sperm, a protein called EFCAB5.
EFCAB5. Okay, so finding it right next to the master builder CFA91 suggests it's also important.
It strongly suggests a connection, yeah, either working together directly or having related functions in that specific location.
So what does EFCP5 do? Is it another builder or something else?
That was the next question. So they did a similar experiment, created knockout mice, this time lacking EFCAB5. Now, these mice were different. They weren't completely infertile like the CFAP91 knockouts.
Oh, okay. So not essential for just having sperm.
Right. They had slightly decreased fertility, but they could still reproduce sometimes. And their sperm looked pretty normal structurally. Tail length was fine, head shape okay.
So EFCAB5 isn't building the basic structure then?
Doesn't seem like it. But here's the key finding. Their sperm had significantly reduced motility. They swam slower.
Ah, so it affects the movement.
Yes. And specifically, they had abnormal movement patterns, particularly during something called capacitation.
Capacitation. That's the process sperm go through to get fully ready to fertilize, right?
Exactly. It involves changes in how they swim, including a kind of hyperactive movement. And certain measures of that movement, like how sharply the tail bends, something called the alpha angle, well, that didn't increase properly in the sperm lacking EFCAB5.
Okay, so let me try an analogy. If CFAP91 is the builder, making sure the house itself is constructed properly.
Yeah.
Is EFCAB5 maybe more like the electrician or the HVAC specialist, fine-tuning how things run inside the house, making sure the systems work optimally?
That's actually a really good analogy.
Yes.
CFAB91 is fundamental for building the structure, the flagellum itself. EFCAB5 seems crucial for controlling the specialized movement of that structure.
Fine-tuning the performance.
Precisely. And interestingly, EFCAB5 seems to be pretty specific to sperm. It plays this unique role there, unlike maybe in other cells with cilia. And researchers have even found that its expression is lower in some men experiencing infertility.
Which really strengthens the link to human fertility issues.
It does. It shows this isn't just a mouse thing. It likely has direct relevance for humans too. It highlights this neat division of labor. One protein builds, the other regulates the movement. Both vital, but different jobs.
So connecting this back to the bigger picture then, these discoveries, identifying CFAP91 as the scaffold and EFCAB5 as the movement regulator, this really deepens our understanding, doesn't it?
Profoundly. It gives us a much clearer view of the actual molecular code that makes sperm work. We're moving beyond just observing sperm under a microscope to understanding the specific molecules involved.
And for you listening, why should you care about these specific protein functions? Well, this isn't just, fascinating science in a lab.
Absolutely not. This knowledge has real potential. It could lead to much better diagnostic tools for male infertility. Imagine being able to test for specific protein defects.
Instead of just a general low motility diagnosis.
Exactly, more precision. And that precision could pave the way for more targeted therapies. Instead of sort of guessing, you could potentially develop treatments aimed at fixing or bypassing a specific molecular problem. It's a big step towards more personalized approaches to fertility.
It's also worth touching on some of the maybe complexities or nuances in the research itself. It wasn't all straightforward, was it?
No, research rarely is. For instance, those initial CFAP91 knockout mice, they didn't just have fertility problems, they also had other issues, like hydrocephalus fluid on the brain.
Oh, right, because CFAP91 probably has roles in other cilia in the body, not just sperm flagella.
Exactly. Cilia are important in lots of places, like the brain ventricles. So the researchers had to do extra work, like breeding the mice onto a different genetic background, to really isolate the effects specific to sperm fertility. It shows the careful work needed to tease these things apart.
And it highlights the difference between the two proteins too, right? CFAP91 affects the actual building of the tail.
Right, which makes it hard to study its role in regulating movement because the tails weren't properly formed to begin with. Whereas EFCAB5, since the culture was mostly okay, clearly showed a role specifically in controlling that movement.
So different proteins, distinct jobs formation versus function regulation.
Precisely. And that detail about EFCK5 being easier to find near CFE-1, specifically in mature sperm, using that proximity labeling, that really underscores the specialized nature of sperm biology. The interactions might be different or more critical in the final functional cell.
So wrapping this up, we've taken quite a deep dive today. We've uncovered how CFAP91 essentially lays the architectural groundwork for the sperm tail.
Acting as that crucial scaffold.
And then EFCAB5 comes in to fine-tune its specialized movements, the swimming patterns needed for that final journey.
Yeah, and understanding these specific roles for both proteins is a really significant step forward. It helps unravel the complex puzzle of male infertility and offers genuine hope for better diagnostics and potentially new treatments down the line.
Ultimately, this research just reminds us of the incredible precision packed into life at the microscopic level. A single cell, engineered with such complexity. And it leaves you wondering, doesn't it, how many more intricate biological codes like this are still out there, waiting for us to decipher, and how will unlocking them continue to change how we think about health, disease, and maybe even the very start of life itself. We hope this deep dive has given you a clearer picture and maybe sparked some more questions. Thanks so much for joining us.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.